Hello, our next speaker is Professor Zheng Zhang from the Shenzhen Third People's Hospital and School of Medicine. In the past half a year, he's one of the early groups to really start to use a single-cell sequencing technology to study the immune cells in COVID-19 infected patients. He has published a really nice paper in Nature Medicine, and he ever since has done additional work. We look forward to his great presentation. Yeah. Okay, go ahead, Dr. Zhang.
Okay. Yeah. And dear friends and dear professors, and it's my great honor to present our recent study here. So my topic is single-cell sequencing reveals the immune pathogenesis in COVID-19 patients. So, and COVID-19 is recognized under the global pandemic. Up to now, there are more than 40 million infections and more than 1 million deaths were caused by the COVID-19. It also there is no effective vaccines and treatment. So, and compared to previously occurred SARS and MERS, the SARS-2 caused more infections, more deaths in more countries and areas. So you also know the host immune responses play an important role in the outcome of the various infections. So as for the COVID-19, most of the people develop into the asymptomatic status, and some of infections progressed into the mild status. Among these mild and moderate status, many of them further developed into the severe, even critical status, and even to death. So, however, we don't know what is determined on the various outcome of the SARS-2 infections. So my first question is, where is the SARS-2? So, and to answer these questions, we should develop a new tools to identify the infected cells, which will aid for better understanding host in various interactions. Although there are several methods to tracing various infected cells in vivo, and in the case of the human clinical samples, these tools are limited, making the passenger infected cells and the viral reservoir cell type hard to detect. So Professor Edo Amit from the Weizmann Institute and I cooperated to develop an unsupervised pipeline for characterization of the viral infections in single-cell RNA-seq. So ViroTracker can map the single-cell RNA-seq data onto a large of the database of known viral genomes. And also ViroTrack can provide annotation of the infected cell type and enables transcriptional sorting of the viral infected cells and versus bystander cells, and even recover the virus induced program and host factors. So up to now, ViroTrack contains the sequence from the nearly 10,000 virus. So then we enrolled 3 mild patients and 6 of the severe patients with COVID-19. And we also enrolled 4 healthy subjects as control. We took the bath as samples to perform the 10x and further do the single-cell RNA-seq and the single-cell T-cell-seq. So mast-cell analysis grouped the mast cells into the myeloid lineage and lymphoid lineage and epicellular lineage, and each lineage was further divided into the smallest subsets. We also found there are some key difference in the composition of the bath samples from mild and severe patients. So, and we applied the viral track onto the data generated from the 9 SARS-2 infected patients. And we can see the SARS-2 transcripts were detected in all 6 of the the severe 6 samples from the severe patients in variable amounts, ranging from the less than 400 to the more than 50,000 transcripts. By contrast, the no viral rates were detected in the 3 mild patients. The coverage analysis further revealed the majority of the viral rates mapped to the 3 ends of the virus segment. So next, we compared the various positive cells and various negative cells. We observed a very strong enrichment of the SARS-2 viral rates in the stellate cells and epicellular progenitor cells. And these 2 cells subsets was known as the cellular targets of the SARS-2 because they express high levels of ACE2 and TMPRSS2. So very interesting, we also observed the enrichment of the SARS-2 rates in the SPP1 positive macrophages, and which suggests that the SARS-2 can infect immune cells or the SPP1 positive macrophages can cytosis viral infected cells or particles. So then we compared the transcriptional expression between the viral positive macrophage and negative macrophages, and we found that as compared to the UMAP negative macrophage, amount of the chemokines such as the CCL7, 8, and the CCL18 was highly expressed by the UMAMI positive macrophages. So, and this section we concluded that first we developed the viral track. This is a computation framework to analyze the host virus infection map. And the viral track can sort infect cells from bystander cells and reveals the virus-induced expressions. And the SARS-2 can infect the epicellular cells and alter the immune landscape in severe patients. So next question is what type of the immune response in the lung ensued after SARS-2 infection. This UMAP finger shows a total of the 32 the clusters according to some typical markers. And in general, these cells could be divided into the macrophages and neutrophils, dendritic cells, NK cells, T cell, B cells, and plasma cells, and epicellular cells. So, and more importantly, we found the macrophage and the neutrophils proportion was increased in the buff from severe patients as compared to the mild patients. By contrast, the MDC, PDC, and the T cells was largely reduced in the severe patients. So this data shows that there is an unbalanced immune cells in the buff of the severe patients. So next, we further focus on the macrophages, and we comprehensive analysis the macrophage subset. So according to some typical markers, and the macrophages can be divided into the FCY, positive the group 1 macrophages, and FCA1 low SPP1 positive group 2 macrophages, and SPP1 positive group 3 macrophages, and FABP4 positive group 4 macrophages. So what is the difference between the functions of these 4 macrophages? So from the table, we can show that the group 1 and the group 2 macrophages are M1-like macrophages, and the group 3 is M2-like macrophages, and the group 4 is similar the alveolar macrophages. Then we found the FCA1 and the SPP1 positive macrophages were significantly increased in the severe BAF as compared to the mild BAF. By contrast, FABP4 positive macrophages was reduced in the severe patients as compared to the mild patients. So this shows that the pro-inflammatory macrophage was largely enriched in the BAF of the severe patients. So finally, we further detected the mRNA expression of the cytokines and the chemokines by BAF. So, and we found the L1 beta, L6, and the TNF alpha was significantly increased in the macrophage from the severe patients, and which was not shown here. And we also found various chemokines CCL2, CCL3, CCL4, and some other chemokines was expressed at high levels of the in lung macrophages from the severe patients. And also we found the CXCL9, CXCL10, CXCL11, which can attract the T cell and NK cells, was much higher in both mild and severe patients as compared to the healthy subjects. So in addition, we also found detected the cytokine and chemokines in the BAF. So we found the severe patients have much higher levels of the inflammatory cytokines, particularly for L1 beta, L6, L8 in the BAF. And very interesting, there are 3 critical cases. The BAF was persistently detected. So we found that these 3 cases displayed the persistent high levels of cytokines. So what happened in the lung tissue in the COVID-19 patients? Here we show the case. This case is a 66 years old man with pre-existing hypertension. So the clinical conditions of the patient progressed, deteriorated after hospitalization, and he eventually succumbed despite of the many attempts to save his life. So we detected the dynamic the SARS-2, and the dynamic monitoring of the SARS-2 shows the various presence in the BAF of the upper airway samples for more than 23 days. Although the SARS-2 is undetectable 17 days later in multiple samples, we still identified the viral RNA in his lung tissues by qPCR, and viral presence further confirmed by the ensued RNA hybridization. Moreover, viral NP and spike protein in the lung were detected by the immunoblotting. The kernel virus-like particles were also identified by the electron microscopy. So this is very interesting to observe the SARS-2 persistent in the lung, although the upper airway samples remained the virus inactive, so which suggests the viral reservoir and ongoing viral activities in the SARS-2 infected lungs. So more important, we also observed obvious lung fibrosis in this patient, as indicated by the cellular threat, muscle staining, and collagen HCA. So which suggests that the severe COVID-19 can cause the lung fibrosis in this case. So to further explore the potential molecular mechanism underlying the lung fibrosis in the severe COVID-19 patients, so we mapped the receptor ligand pairs among the different cell subsets to construct a cell-to-cell interaction network. So the center of the interaction network in control was our endocellular cells and the fibroblasts, so which is likely a requirement for the normal lung homeostasis. By contrast, the macrophage and the fibroblasts move to the center in the COVID-19 patient, so which suggests that the macrophage-derived signals might play an important role in the shaping the patient lung microenvironment. So the analysis further shows the CXCL12 highly expressed in the patient fibroblast, which might enhance the increment of the multiple immune cells. And in addition, macrophage produce more 1-beta and TGF-beta to moderate the fibroblast functions in this patient. So in it, we found that the macrophage produce more 1-beta and TGF-beta, and in it, the patient fibroblast shows the activated phenotype and increased the expression of extracellular matrix such as the collagens. So taking together, our data highlights the crosstalk between the macrophage and the fibroblast through the well-known I1-beta and the TGF-beta signal pathway as the underlying driving for the lung fibroblast in the severe COVID-19. So for this section, we conclude the single-cell landscape of the bronchial immune cells in COVID-19 patients reviewed by the single-cell RNA-seq. And we also observe the pro-inflammatory monocyte-derived macrophage was abundant in the BAF from the severe COVID-19 patients. In our case, we found a persistent viral activity, cytokine storm, and lung fibrosis. So here. So then we analyze the TCR clone type within the T cells from the COVID-19 patients. So these cells, T cells can be divided into the 7 subsets according to these typical markers. So very interesting, we found the clonal expanded CD8 T cells show the aggregative distribution with the transcriptional homogeneity. And while the clonal expanded CD8 T cells from the severe patients show the diffusive distribution with the transcriptional heterogeneity. Then we take the property of this defined highly clonal expanded CD8 T cells in the mild patients. They show that there is a significant increase in the tissue resin markers for this expanded CD8 T cells from the mild patients. So this shows that the highly expanded CD8 T cells may actively participate in the controlling of the SARS-2 infection. So we conclude that CD8 T cells in severe patients BAF was less expanded, more proliferative, and more phenotypical heterogeneous. By contrast, there are large proportion of the CD8 T cell effectors with C2 resident and highly expanded features were present in the mild patient BAF. So here we propose a model to describe the immune landscape of BAF in SARS-2 infections. So, and as a model, in the mild patients after SARS-2 infection, the lung resident CAT cells was clonally expanded, which may link to the virus clearance. By contrast, in the severe patients after SARS-2 infection, and the effector cells can produce a amount of the chemokines, which further recruit more pro-inflammatory monocyte neutrophils and some other CTLs into the lung, and which leading to the inflammatory injury. So the third question is what's the difference of the immune response between the lung and the peripheral blood in SARS-2 infections? Here we pair the PBMC and BAF and perform the single-cell RNA-seq and the T-cell-seq. We wanted to answer two questions. The first is the difference between the systematic and the local immune response during SARS-2 infections. The second is whether cell migration from the peripheral blood into the lung during the SARS-2 infections. So first, let's say the peripheral blood immune cells. So, and in the peripheral blood, the immune cells could be divided into the T cells, B cells, monocytes, and some other cells. So, as compared to the mild patients, the severe patients displayed the increase of the monocytes, especially for the CD14 positive monocytes, and the decrease of the T cells and NK cells. Then we focus on the myeloid cells in peripheral blood. We found so we compared and because the CD14 positive monocytes was largely increased in the severe patients as compared to the mild patients. So we compared the differential expression of the transcripts in CD14 monocytes in the three groups of the subjects. And it is found that the interfer pathway was both increased in the mild and the severe patients as compared to the healthy subjects. But we found the neutrophil activation pathway was peripherally enriched in the monocytes from the severe patients, but not mild patients. And especially the S100A8, A9, and A12 was significantly upregulated in the monocytes from the severe patients. So by contrast, the type 5 interfer and antigen processing and presentation pathway was significantly reduced in the monocytes from the severe patients. And for example, the MRC2 molecules and the cytokines. So this data shows the remote the monocytes of the peripheral blood in the severe COVID-19 patients. So we further set up the characterization score and the MRC2 score and found the monocytes from the severe patients displayed the high levels of the characterization score and the low levels of the MHC2 scores. We further confirmed the decreased HLA-DR by peripheral monocytes in severe patients. In addition, this co-signature score was found to positive related plasma inflammation levels and negative with the peripheral T cell counts. So this data shows that the peripheral monocytes displayed the MDIC-like or immature monocyte status, which was involved in the immune paralysis in severe patients. Then we compared the
just 1 minute, Dr. Zhang. Maybe just 1 minute.
Okay, and I will be quick. So here we compared the peripheral and the BAF monocytes, and we found the peripheral monocytes display the MDSA-like by contrast in the lung as beta positive inflammatory monocytes. So, and this is the peripheral T cells. And the T cell sharing analysis, there is a peripheral activation of peripheral CD4 T cell response in the COVID-19 patients. And also we found that there is an active recruitment of the peripheral CD8 T cells into the lung in severe patients. So this is concluded for this sections. Okay, and we thank our team and our operators. So thank you for your attention.
Thank you so much. So let's see, we have questions. So far, we don't see a lot of questions, but let me ask you a question. I think I previously had a similar kind of discussion with you. So in your different part of the presentation, you talked about the elevated expression of certain cytokines. In some cases, you also mentioned the overexpressions of S100A9 and A10. So such increased expression can be explained in, well, two ways. One is the same cells will somehow turn on their gene expression for chemokines, like CCL8 or some other genes. The second model, though, is not that the cell that turn on the expression, is that the population have a different mixture of different cells. The new cells already have higher expression of those genes. So in that case, you're arguing for not the upregulation, but just recruitment of additional cell types. So in your opinion, which one is more likely?
I think the latter maybe is a major reason. And I think the maybe monocytes from peripheral blood include was recruited into the lungs and where and maybe tend to the inflammatory and macrophages. And this macrophage can produce the high levels of the chemokines.
All right. Yeah. Okay. Also, you did mention that S100A9, A10 genes are upregulated, but based on the heat map you showed, it seems like their expression level actually pretty consistent in healthy, mild, and severe symptoms patients. Actually, I don't know. Maybe it's a visualization thing, but I don't know if you recall such results where the S100A9, A10 genes. You did highlight that the genes show upregulation, even though the picture shows fairly consistently high expression.
You mean the peripheral blood, the monocytes can persistent expression the S1?
The macrophages. Right.
Yeah. And so and maybe yeah, and now and we are doing detect continuously to detect these cells, but this data have not shown here.
Okay.
And, yeah.
All right. Yeah, great. Yeah, I think we're running short of time, so I want to thank you again for those for generating really important data sets and those nice findings. Thank you very much.
Okay, thank you.